Can you hear me? So, hello everybody. Welcome to first Mendel lecture of this season. And as you know, this is a part of our Life Science Seminar series, so it's considered like the VIP part of the seminars. And it's a pleasure for me to introduce our today's speaker, which is Professor Erich Nigg from University of Basel. So Erich is actually director of the largest department called Biozentrum at the University of Basel, and I can say a few words about his career and research. So he's a Swiss, and he did his PhD at ETH in Zürich, working on biochemical and biophysical properties of membranes, and then actually moved, or probably decided to look what's behind the membrane. So he went for a postdoc to University of California, San Diego, where he studied different aspects of cancer cell lines. And then he returned back to Switzerland, first to ETH, and then he spent some time on ISREC, which is a Swiss Cancer Institute, and eventually ended up in Geneva. And during the time, he was actually again changed. His main interest, so his first work was based on proteins in the nuclear lamina. He was interested in a lot in transport between cytoplasm and nucleus. But as he was moving from Zurich to Geneva, he was also gradually moving more and more into kinases, and eventually ended up studying various aspects of cell cycle. And then again, he moved when he was appointed the director of Max Planck Institute of Biochemistry in Munich. And at this time, he fully engaged on the regulation of cell cycle from the perspective of control of mitosis. And also, it was a time where he became more and more interested in an organelle called Centrosome, which I'm sure you will hear about from Erich later on from his talk much more. And maybe 8 years ago, he decided to kind of finish the circle, and he came back to Switzerland at the University of Basel, where he's a director of the Biozentrum department. So I would say, to kind of describe Erich's journey over the world and with the science, is that he moved from, he not only moved from, different universities and different countries, but he also moved from different topics. And I would say that any topic he kind of touched on, he was able to kind of really contribute with the seminal discoveries, which I think is quite unusual, given that, a lot of people tend to really stick to one particular subject and try to really get specialized. This is not how Erich felt. He always was looking for new challenges. And so, once again, I'm very glad that Erich accepted the invitation to have the first Mendel lecture here. And before I give him the microphone, there's one more thing I'm glad I was tasked to, and this is to award Erich with a Mendel medal. So thank you very much for coming. It's there.
Wow, okay, yeah.
And so now the stage is yours, and we're looking for your talk.
So I'd like to thank Lukas and the organizers for inviting me. I'm really impressed by the location, and I'm greatly honored to give a Mendel lecture. You can tell how honored I am from the fact that I wear a tie, which actually happens rarely. Now, Mendel, of course, is one of the greatest icons of science that we can think of. But Mendel actually did not know about chromosomes because this was after his time. Conversely, this other hero here, Walter Fleming, who described mitosis, he did not know about genetics. So he described in I find amazing detail in 1882, how chromosomes condense, how a mitotic spindle apparatus forms, how the chromosomes align in the spindle. You see here actually he understands sister chromatids, how they get pulled apart, how a central spindle form, how the cell divides. He described this all in amazing detail, but he did not quite know what he was looking at. And I think this should be a lecture of humility also for us. I always wonder how many things we look at and we don't really understand what we are looking at, because there's no reason to think that times have changed that fundamentally, that now we understand everything. Far from it. But we now can buy fancy equipment and smart software, and we have labels for different structures in the cell, and so we can make believe that mitosis occurs in color. And you see it here. Beautiful cell division, time-lapse movies. There are two cells here by chance. Look at this one first. It starts and is very efficient, very professional about the process. Look about this one. It starts before the other one, but it has a hard time capturing this chromosome here. And the whole cell waits until this last chromosome is captured. And that's because there's a surveillance mechanism known as a spindle assembly checkpoint that makes sure that cell division does not occur, mitosis does not complete until everybody is on board. And that is very important to prevent chromosome missegregation. Now, I must confess, when I was asked for a title for this lecture some time ago, I gave a kind of a sweeping title, and I thought I might want to talk about some of this work we've done in this field, but then kind of a year went by, and when I put together my slides to come here, I had no longer in mind the title that I had given a year ago. So my talk will not quite match the title, but I at least will spend a little bit of time on kinetochores and chromosome segregation. But the bulk of my time I'll devote some of our most recent work. Some of it is unpublished about the centrosome cycle, which I also think is very interesting. Anyway, you'll bear with me. So mitosis is not just a beautiful process, it's also an important process because if you look in cancer cells, for example, you always see, or very commonly see, major chromosome rearrangements. You see both numerical aberrations, aberrant numbers of chromosomes. You also see structural aberrations where chromosomes are broken and fused together. This is a very characteristic feature of many different types of tumors. And generally, the more aggressive the tumor, the more scrambled the genome. Here is a normal genome for control. Now, when you think about how these features come about, or how they are likely to come about, and particularly if you focus on whole chromosome number aberrations, there are really two processes that you will tend to focus on. One is chromosome segregation per se, so the process of M phase, of how you attach chromosomes on the spindle apparatus and how you pull them apart faithfully. And the second is the centrosome duplication cycle, because the numbers of centrosomes you have in the cell, this is at least true for animal cells, determines the efficacy and the fidelity of chromosome segregation, because you want to have a bipolar spindle, so you want to have only two centrosomes, one at each pole. So today, because I had promised in my early title I would say something about kinetochores, I will fulfill my contract and say something about kinetochores, but I'll keep it very short, show you essentially no data, just give you a little bit of a take-home message, and then I'll move on into the second part. So the first part really addresses the key question, and I'm not saying that we're going to answer it, but the work is related to this key question. That is, how do cells optimize microtubule attachment to kinetochores so that chromosome segregation occurs in an error-free fashion? Now, kinetochores, as I'm sure you know, are those specialized proteinaceous structures that form on a specialized DNA structure known as the centromere, which in humans is composed of alpha satellite DNA. And you can visualize this, for example, by staining with antibodies against SEMP-PA, which is a specialized, an isoform of histone H3. So this is this red staining here. But there are probably a couple hundred proteins that make up this proteinaceous platform into which microtubules insert, and they have to insert from the two poles, one into each sister chromatid, kinetochore, so that you can then pull them apart. So what the cell wants to achieve for every one of the chromosomes in the cell is that they are attached in this manner by orientation or amphitelic attachment. This is what you want to achieve. Inevitably, in the process of achieving this, the cell goes through stages where only a single kinetochore is attached but the other is not, or whether both kinetochores are attached to microtubules from the same pole, or worst of all, where the same kinetochore is attached to microtubules coming from both poles. All these things happen, but these errors here should be corrected, ideally, so that all chromosomes are in this configuration before sister chromatids are allowed to separate and be pulled apart. Now, how does this work? We don't understand this for sure. The world doesn't understand it fully, but here is some current thinking. One is that error correction relies heavily on a protein kinase known as Aurora B, and a consistent finding is that if you are in a situation where the attachment is not yet achieved the way it should be, the kinetochore microtubule interaction is unstable, so there's a high turnover here. But once the microtubules from both poles attach properly to the kinetochores on the two sister chromatids, then there is tension that develops. That is because cohesin proteins keep the sister chromatids connected and resist the forces that the microtubules, the pulling forces the microtubules exert, and so you stretch the kinetochore. And so one model that has been proposed quite some time ago is what is known as a dog on the leash model, and I like it a lot because I like dogs. This is Vasco, my dog. The idea is that the dog sits here, Aurora B is the dog, and it sits here at the center, and it phosphorylates proteins that are mediating the microtubule interaction. And by phosphorylating them, it releases this interaction. It lets the microtubules let go again. And the idea is that once you have the tension developing and the kinetochore stretches, now these critical targets get out of the reach of Aurora B kinase. Like the dog can no longer bite if you are sufficiently far away because the leash is too short. That's the idea. It's a nice idea. I like it. It's attractive but not necessarily correct. So I do not want you to go home and think we understand kinetochore microtubule interactions on the basis of this model. What we do believe is established is that Aurora B does destabilize constantly inappropriate kinetochore microtubule interactions so that the cell can try again to get it right. And only when it gets it right, these interactions become stable. By some mechanism we do not fully understand. So the open question is, what features allow Aurora B to distinguish proper bipolar attachment from inappropriate kinetochore microtubule attachment? And so we've made a little bit of a contribution to this, I believe. That is work that grew out of some ambitious projects we started when I was still in Martinsried in Munich. And one of the advantages of a Max Planck position is you have a lot of firepower. You can do big projects. And so we decided at the time to purify spindle apparatus from human cells, from HeLa cells, and then use mass spectrometry to find out as much as we could about the components in the spindle apparatus and also the phosphorylation sites in the spindle apparatus. And one of the perhaps most rewarding things that came out of this was we discovered a protein complex which we call the SCA complex for spindle and kinetochore associated. And you see antibodies raised against these proteins, which originally we discovered SCA1 and SCA2, and then a few years later we and others found SCA3, and these three proteins, they really form a 1-to-1-to-1 stoichiometric complex. And all three of them, you see, it is red staining here, beautifully localized to kinetochores. Now, when we looked at the function of these proteins, of course, we did RNAi experiments, so we depleted these proteins. And here you see now cells dividing. These are histone H2B labeled cells, so the chromosomes are in green. Here is the control. You see very efficient metaphase plate formation and separation. When we deplete SCA1, we see what we think is a hypomorphic phenotype. So the plates form, but then they wiggle around for quite a while until eventually they separate. And when we deplete two of the three components of the complex and produce a more severe phenotype, then we see chromosomes still condense, but they never really form a proper metaphase plate. So somehow the SCA complex is required for proper alignment, for capture of chromosomes by microtubules. Now, prior to the discovery of the SCA complex, people thought about kinetochore microtubule interactions in the following way. There's a so-called KMN network. KMN stands for the names of proteins in three different subcomplexes, and I don't want to go into any details. But the idea was that somehow this KMN network grabs the microtubule in a way that leaves the tip of the microtubule free. And that is important because microtubule must polymerize and depolymerize all the time to move chromosomes around. So it is like a ratchet that holds on like this to the microtubule. It's not like a stick that you glue to a plate. You leave the end free that it can grow and shrink. So that's what the KMN network does, and it was also known that Aurora B phosphorylates several of the subunits of the KMN network. Having discovered the SCAR complex, we of course asked, well, how does this fit into the picture? What role, if any, does the SCAR complex have here? And I'm just cutting short, not showing you any of the data, but basically what we found is that the SCAR complex components interact directly, multiple subunits with multiple subunits of KMN network, and together these interactions are required to stabilize end-on microtubule kinetochore interactions. And we also found that Aurora B kinase phosphorylates not only KMN network components, but also SCAR complex components. And by phosphorylating these components, it tends to knock them off the kinetochore all the time. So that's part of how Aurora B destabilizes kinetochore microtubule interactions. Then we've done some collaborative work with Elena Conti's lab at the time in Martinsried and with Arokya Jayaprakash, who is now in Edinburgh, and found that the SCAR complex forms, through X-ray crystallography, forms this W-shaped molecule and probably associates something like this with the KMN components and the microtubules, and we believe that this helps the microtubule remaining attached while the microtubule grows and shrinks. Now, more recently, Patrick Redley, who was one of my last PhD students, did some quite amazing work, actually, and found something really unexpected, and that is that the SCAR complex not only is a major substrate of Aurora B kinase, but also activates Aurora B, promotes Aurora B activity. Now, that creates somewhat of a conundrum. Now you have a complex which needs to be phosphorylated or is phosphorylated by a kinase and thereby removed from the kinetochore, and at the same time it stimulates the kinase that removes it. So how can it ever accumulate under these circumstances? Now we think that this feedback loop that we have, where SCAR complex activates Aurora B, but Aurora B inhibits, so to speak, the SCAR complex, this is a very important feedback loop. And we believe it helps to achieve the following. If you think about the challenge the cell faces when it attaches microtubules to kinetochores in order to separate chromosomes faithfully, it really has to find a sweet spot of attachment strength or turnover dynamics. Because if the turnover is too high and you have an insufficient kinetochore microtubule occupancy, then you can never satisfy this so-called spindle assembly checkpoint, which I illustrated on my early movie, where when a chromosome is not properly attached, the cell will not move forward. It'll wait. And so if you depolymerize all the time and you always lose these attachments, then you'll never get beyond this spindle checkpoint. So you're stuck in mitosis forever. Conversely, if you have too tight interactions between, too strong interactions between kinetochore and microtubules, then whatever error correction system you envision will eventually be overruled, and you will have an accumulation of these malorientations, which are known as merotelic attachments, and this will then lead to chromosomes missegregating because they can't decide which way to go if they are attached to two poles. So you really need just the right attachment strength, and we believe that this feedback loop may well be at the basis to find the right attachment strength. And we believe what changes between a situation before you have achieved bipolar attachment and once you have achieved bipolar attachment is that, not depicted here, but is that phosphatases come in, because phosphatases are brought in by the KMN components and they will tilt the balance then in favor of the SCAR complex against Aurora B. So this is just some food for thought, and if you want to read up on this, I gave you the references where you can find them easily in PubMed. So that was it for today for kinetochores and microtubules and chromosome segregation. And I'll now move on to my other major love, which is the centrosomes. Here is a centrosome, seen as one of the early slides, actually, that Theodore Boveri, one of my other heroes, has prepared, and Joe Gall in the United States has gotten a hold of these slides. These were prepared in Würzburg at the time. And then after the war, they ended up in the United States, but they're now back in Würzburg. And this picture is taken from one of these original slides. And you see here a centrosome and a centrosome. That's what Boveri first saw. And because these pictures have been widely publicized, many people think of the centrosome as a division organelle, as a mitotic spindle pole organelle. But that's really a misconception because we have always known that higher plants, land plants, don't have centrosomes, and yet they make bipolar spindles. Likewise, eggs of animals, many animals don't make meiotic spindles that have no centrosomes. So you don't need centrosomes to make bipolar spindles. But when you have them, yes, then they are very important at the spindle pole. Within the centrosome, and this Boveri could not see at his time, there are two cylindrical structures known as centrioles. These are microtubule-based cylinders with some bells and whistles, and they are embedded in a pericentriolar, in a matrix of proteins known as the pericentriolar matrix. So one centrosome comprises two centrioles. Now it turns out that the older of the two centrioles here actually has another function, and that is it forms the basal body for the formation of cilia and flagella. And evolutionarily speaking, this is probably, almost certainly, what was selected for. So if you look across all species, whenever a single cell type in the life cycle of a species forms a cilium or a flagellum, then this species is going to have a basal body. So this is a very strong indicator that this is the function which has been selected for. But as I said before, this spindle organization during mitosis is not the only function of the centrosome because it also organizes microtubule arrays throughout interphase of the cell cycle. And this in turn is important for positioning of organelles, it's important for motility, it's important for polarity. So the centrosome has much more to do than mitosis. Still, here is now the picture of Theodor Boveri. He launched at the time another very important concept, or concept we're still working on, in a famous book he published shortly before his death, and he called On the Origin of Malignant Tumors. And he apparently did experiments where he did dispermic fertilizations and realized that whenever you have apparent numbers of centrosomes in your cells, then it's very hard, almost impossible, to correctly segregate chromosomes. And apparently he talked to pathologists, and they told him that such kinds of multipolar spindles were a very characteristic feature of what they saw in the hospital when they sliced human tumors. And so he put two and two together and postulated that perhaps centrosome aberrations are actually causing chromosome missegregation, and that would cause tumorigenesis. And I'll return to this concept towards the end of my talk. I should say this is still at the stage of hypothesis, even after more than 100 years. There's no strict genetic proof that would say this is what's happening. This being said, there is irrefutable genetic evidence to link centrosomes and centrioles to other human diseases. On the one hand, there are diseases now known as, or collectively referred to as ciliopathies. There are some prominent phenotypes depicted here, polycystic kidneys, polydactyly, and obesity. There are a whole number or a whole range of different syndromes, different names. Some of these diseases are very severe, others are very mild, but most of them are quite severe. So this is kind of easy to rationalize. If you have a mutation in a critical component building the basal body, then you can properly build a functional cilium, and that will give you this pleiotropic disease. What I think is a priori more difficult to rationalize is the other disease which has emerged, which is microcephaly and dwarfism. Turns out that many cases of inherited diseases of this type, when the genes were identified, they were found to code for centrosomal proteins. So then you wonder, how is it that a protein or a gene that codes for a protein at this small, tiny organelle, important for microtubule organization, if you mutate this, it gives you a small brain or a small body, and that is a question. So I'll run through three chapters now in my little book here. The first is I want to just give you some basic background information on the centriole duplication cycle, give you a cast of characters, introduce the main components that are important for this, and then I'll address this question of how centriole amplification could contribute to primary microcephaly, and finally I'll return to the question of whether centrosome aberrations really have something to do or could have something to do with cancer. So here is the challenge that the cell faces every time it wants to prepare for a mitosis. It starts out in G1 phase of the cell cycle with one centrosome comprising two centrioles. This one has the appendages, this one can form cilia. Before the cell goes into mitosis, you have to duplicate everything. So you have to have two pairs of centrioles. Now, I think if you were an engineer and you wanted to duplicate something like this, what you would do is you would grow it to twice the length and cleave it in the middle. At least that's the way I would do it, kind of like fission yeast. Right? But the way nature does it is it grows a new cylinder at an orthogonal angle next to the pre-existing cylinder, which immediately raises very intriguing questions. Why is it only one? Why don't you form multiple ones? What determines the length, and so forth, and so on. So this is from a cell biologist's perspective, is very intriguing questions here. Now, I think it's fair to say that the first really molecular insight into what is going on, we owe this organism here, Caenorhabditis elegans, and the people that have been working on this. Through genetics and reverse genetics, they actually came up with only a handful of genes that were found to be specifically required for centriole duplication in the worm. And then for a while it was quite frustrating because nothing could be found outside of nematodes that seemed even remotely homologous to this. But eventually the dust has settled, and now we know that all of these components have counterparts in Homo sapiens. So SIG1 turns out to be PLK4. They are quite distantly related, not obviously recognizable homologs, but we are all convinced that these are functional homologs. Then you have a bunch of coil proteins here. I'll just now say a few words about three of these components because they're the ones which I think are attracting currently most attention. PLK4, STIL, and Sus6. What these three proteins have in common is that when you deplete the corresponding messenger, then centriole duplication stops and you run out of centrioles over proliferation. When you overexpress any one of these, you form multiple centrioles at the same time. So all three are clearly critical duplication factors. Now, PLK4 was discovered both in my lab and independently by Monica Betancourt-Diaz in Drosophila in David Glover's lab more than 10 years ago, and is now the master regulator of centriole duplication. At the time, we began to work on this simply because it resembled another kinase we had long been working on, namely polo-like kinase 1. And this was kind of an orphan member of the family that nobody had ever looked at. And so when I had a PhD student, he began to look at this and found that this thing beautifully localizes to centrioles. And not only, as we deplete this protein by RNAi, we see that we run out of centrioles. And conversely, when we overexpress it, we simultaneously form now multiple centrioles. You see here the mother seen from top to bottom. Here is PLK4 in red. And now, rather than building just one centriole at an orthogonal angle, we build 4 or 5 at the same time. So PLK4 is the master regulator. Sus6 for a long time looked like just a boring coil protein. Nobody could recognize anything interesting about it until two laboratories, both Pierre Gönchi and Steinmetz in Lausanne and Van Breugel and co-workers in London, actually began to do structural work. And to interpret their structural work, you have to understand the following, and that is, it has long been shown, primarily by through work in Chlamydomonas, that the first thing which is built when a new centriole is built is a so-called cartwheel. Here you see a cartoon of this cartwheel. It's named cartwheel because it looks like a cartwheel. And this is now a real cryotomography image of a cartwheel from this species here, Trichonympha. This is a protozoan parasite that lives in the gut of termites. Nice. The reason why people work on this is because it's full of cartwheels. It has a huge stack of cartwheels, hundreds of them. So this structure here is repeated hundreds of times. This makes it, of course, very amenable to cryo-EM tomography. But the point is, you have to know about this. So you have to find literature which describes these things, and you have to go back to the Abbey, probably library, to find such hidden treasures. Here you see the cartwheel. This was early on, as I said, Chlamydomonas. You see this is from work in Hirono's lab in Japan. Immunogold labeling for the Chlamydomonas version of SAS6. You see beautiful immunogold staining of here the cartwheel seen in cross-section or in longitudinal section. And here you see the cross-section. You see this hub here and the immunogold labeling and the 9 spokes. So this is a real EM picture with SAS6. And now comes structure. So what both Gönchi, Steinmetz and Van Breugel and co-workers have done is they have taken a piece of this SAS6 from Chlamydomonas and then crystallized it. And it looked like this. And then they found there are interactions, and if they put them together in silico, they end up with this. A ring with a diameter that matches this diameter and 9 spokes sticking out just like this. And that this is not an in silico artifact or sheer fantasy. Here is rotatory shadowing electron microscopy. You see reconstituted SAS6 in vitro, ring and spokes sticking out. And this actually has gone further. In the meantime, they are now building slowly, reconstituting the whole cartwheel from purified components. Beautiful work. So this is SAS6. Finally, still. Still is also interesting. Still was first discovered and named because of a rearrangement commonly seen in T-cell acute lymphoblastic leukemia. And people got interested because of this. They knocked it out. It's embryonic lethal in the mouse. There's also zebrafish mutant that has a lethal phenotype. But what this thing is doing was actually largely obscure for a long time. Evidence was building that might have something to do with mitosis, perhaps with centrosome. But the breakthrough came when Jordan Suraf's laboratory at the time in Cambridge, now in Oxford, they found a Drosophila protein they named ANA2 that was important for central duplication in Drosophila, and that looked very much like, looked a little bit like SAS5. And SAS5 had been the last unresolved issue from the original collection of C. elegans mutants that were recognized to be critical for central duplication. So nobody had found at that time a homologue of SAS5 anywhere. And so ANA2 looked a bit like SAS5. It focused attention on a motif called the stun motif, and armed with this knowledge, then Jordan and colleagues went into the human database and lo and behold pulled out human still. It has 3 times the size of these guys, but it looked from the sequence as if this was a legitimate candidate for being the long sought after human homologue of still. And so when Christian Arquint walked into my lab as a new PhD student, he got the project proposed to look at human still, and sure enough, he could quickly show that indeed still is such an essential duplication factor, because normally you have it here sitting at the centrioles, at the spindle poles, but if you reduce still levels, then cells can't duplicate their centrioles, and if you overexpress it, you make extra copies of centrioles. And in the meantime, we and others have been able to come up with the following simple model, oversimplified. So we know now that still binds PLK4 directly. It stabilizes. This is the master kinase that causes centriole duplication. It stabilizes PLK4. At the same time, PLK4 phosphorylates still in the stun motif. And once still is phosphorylated, then SAS6 gets recruited, and we believe then you see cartwheel formation. So this is a beginning of an understanding of a molecular pathway of how centriole duplication is controlled. That's all I want to tell you about this, and now this arms you, or this prepares you for hopefully understanding how we believe centriole amplification might play a role in causing primary microcephaly. Here again, this is Christian, the person who characterized still in my laboratory. And I've told you, levels of still are critical, because if you don't have the right levels, you either run out of centrioles or you have too many of them. So that prompted Christian to figure out how still levels are regulated in the cell. And to do this, he prepared a cell line where still is GFP labeled. If you look over here, it's just about a 1-to-1. This is the GFP form over the endogenous. This does not yet cause centrosome amplification. Now you see, still gets more and more abundant at centrosomes until cells approach mitosis, and then it separates, and then it goes away from the spindle pole first, and then disappears from the whole cell body altogether, and then after division, it comes back into two daughter cells, as you can see here. Christian then figured out what was going on and showed that what happens is the disappearance of still from the centrosome. The first step is triggered by CDK1 cyclin B, the key regulator of mitosis, the kinase, phosphorylating still, we believe. And then the second step is still disappears from the cytosol, and this, Christian could show, is dependent on the activity of anaphase promoting complex / cyclosome. This is the ubiquitin ligase that is critical for cleaving the cohesins when sister chromosomes are separated in mitosis. So this is a major ubiquitin ligase in mitosis. Now, APC is known to degrade proteins provided that they have particular destruction motifs. They come in two flavors. They're called D-boxes or Ken boxes. And so Christian looked at the presence of D-boxes and Ken boxes in the still sequence and found several D-boxes and one Ken box. He then looked at what they might be doing. He mutated them all and found that only one of these boxes, namely the Ken box, causes stabilization of still. So the argument is APC degrades still by recognizing this Ken box as a destruction motif. Now it's where it gets interesting with regard to microcephaly. I told you that several genes that have been mapped and are held responsible for microcephaly, they all code for, or most of them code for, centrosomal protein, and one of them, MCPH7, is still. Now, the first two mutations ever described in patients, these are patients in Pakistan, they have mutations towards the end of the protein, and as a result of these mutations, they get stop codons right in front of the Ken motif. So these patients essentially express full-length still, except they don't have the Ken box. So we tried to get patient material, but this proved to be impossible from Pakistan. And so what Christian did was he engineered these mutations into the still cDNA and put this back into the cell line that I've shown you before, or similarly the same locus where I've shown you before for the GFP movie, where we had just a little bit of extra still, and this did not cause any centrosome amplification. But now if we put in this non-destructible still, and that it is non-destructible, you can see here. So these Ken box mutants, in contrast to the wild type, which gets destroyed by APC, these guys are pretty stable. And as a result of this stability, now in successive rounds of cell division, still protein levels accumulate until eventually you get centrosome amplification, and this increases over time. And so to put this into cartoon form, what we believe is going on in a normal cell cycle is that early in mitosis, CDK1 cyclin B removes still from the centrioles, and then APC removes still altogether through degrading it thanks to this Ken box. But if you're microcephaly mutant, then you don't have this Ken box, and APC cannot degrade still. So in the normal situation, still levels get reset to 0 every time cells divide. But in the microcephaly situation, this resetting to 0 does not occur. And so still levels accumulate progressively until eventually you cause centrosome amplification. Which of course then begs the question, could this centrosome amplification represent a root cause of microcephaly? Is this really the cause of the disease? Now, we cannot claim that this is the case, but what we can say is that there is kind of supportive evidence, and this comes from work done in Renata Basto's laboratory in Curie. What she was working on at the time was she used PLK4, the master regulator kinase that I have introduced, trying to cause brain tumors in mice. So she overexpressed active PLK4 in the brain, knowing she would cause centrosome amplification, and she was hoping to confirm Boveri and see brain tumors. And instead, what she saw was a microcephaly-like phenotype. So what we can say now is clearly still is a central duplication factor critical for brain development. That is genetically solid. We can say that certain MCPH mutations in still do cause still stabilization and central amplification. And in light of the results showing that central amplification induced by another route can produce microcephaly-like phenotype in mouse, we believe it's at least possible to speculate or propose that central amplification may be one prominent root cause of microcephaly. And the answer is simple. The interpretation is simple. You simply kill off the cells at the time when the brain needs to make lots of cells during development. And if you can't make these cells in a sufficient time window, then you get a small brain. Which brings me to the last part of my talk. Cancer, centrosomes and cancer. I introduced this slide before. Theodore Boveri had this idea that maybe you get missegregation of chromosomes and tumorigenesis. Now, in the years since Boveri, people have gone back and stained dozens and dozens of tumors with antibodies against centrosomal components, and they fully confirm what Boveri had thought, and that is that you see very often numerical centrosome aberrations in tumors. These are all breast cancers taken from the literature here. Numerical aberrations, but you also see very common structural aberrations. Massively enlarged centrosome-like structures in these tumors, and oftentimes they coexist. Now, these aberrations have been extensively studied. They cause cell death, not surprisingly. They cause increased chromosomal segregation in line with Boveri's idea. There's other mechanisms have also been considered. But in contrast, structural aberrations have barely been studied at all. People just note they exist, but nobody has addressed them. So I got two postdocs into the lab. Dominique Schnarch, he actually was a medical doctor by training, has now left the lab. And what I'm going to show you now is mostly the work of Olivier Gagné, who followed up on a setup that Dominique had introduced in the lab. And so we decided to look at the consequences of these structural aberrations. What we tried to use is we fabricate enlarged centrosomes in a tissue culture model, 2D and 3D, as you will see. And these enlarged centrosomes, they arise when we overexpress a protein that we characterized many years ago and we called NLP, for 9A-like protein. We know that these blobs very much resemble the blobs that you see in tumors. They also have similar properties in both these guys and these guys, they recruit gamma tubulin, important for microtubule nucleation, and centrin and so forth. So they have a lot of common. Here is a control we often use, CEP68. When we overexpress this protein, it's just diffuse all over the place. Another reason why we focus on NLP is that, according at least to this lab in China, they have shown that this gene is often amplified in human tumors, and when engineered into mice, in transgenic mice, it causes tumors. So there is evidence to think that NLP overexpression is actually occurring in tumors and may be causally related to tumors. So what Dominic set up was he set up this 3D spheroid model where you grow cells in a matrigel preparation, which is mimicking a matrix, basal lamina type situation, a widely used model, and these are kind of normal breast epithelial cells, and we let these cells go through morphogenesis, so we get these spheroids, or mammospheres as they are also called, and then we induce transgenes with doxycycline. So we have transfected these before with retroviruses to allow doxycycline-inducible expression of transgene. And briefly, what we found and published early on was that when we do this with NLP, and we get these aberrations, we disrupt microtubule organization and epithelial cell polarity. We interfere with the disposition of adhesion protein and junction proteins, and also we see enhanced response to growth factor stimulation, but we believe all of this are late consequences. More recently, and this is quite specific here, is, for example, overexpression of SAS6. These are numerical aberrations inducing change. We see none of this happening. More recently, we've looked earlier, and now we see something which we believe is potentially very interesting. It's a very robust phenomenon. We see it in about 30% of all the spheroids. We see it both in MCF10A derived spheroids as well as in MDCK cysts, which is a somewhat different model, also widely used. We see this kind of budding of cells. Now, the first thought is, well, this is just garbage that the spheroid pushes out. Who cares? But fact is, most of these budding cells are alive. 65% of them are alive, at least as judged by absence of apoptosis markers. And secondly, budding relates to mitosis. So in a very heroic effort, and this is not easy because you don't know where it's going to happen. So you film these spheres hoping you catch somebody in the act. And that's what Olivier did. And he filmed so far 35 budding events, and each and every time it's a mitotic cell. Look here. Here is undergoing mitosis. And now it pops out. Look here. Mitosis. Now it pops out. It's quite alive because it's going to divide again. Now. Voila. And then it even moves away, as you can see. All right? So, why is that? Why do structural centrosome aberrations push mitotic cells out of the sphere? Well, first of all, we see that spindle orientation is randomized. Normally in an epithelium you divide in this way, but now we see these spindles are rotating when we have these structural centrosome aberrations. Why do they rotate? Because E-cadherin is messed up. Now, E-cadherin is known to be rearranged every time cells go through mitosis, and I'm not going into great detail here, but we can just say this happens. We see major changes in E-cadherin dynamics. Now, we see these changes in E-cadherin dynamics regardless of whether we overexpress NLP and cause a structural aberration, or whether we overexpress PLK4 and cause a numerical aberration. Yet, only with this with the NLP, we see the budding. We don't see budding with PLK4. And I'll come to this in a minute. So the way we think this works is, and this is again following up on what other people have done before, it's known that if you have enhanced microtubule stability, you activate RAC1, which in turn causes activation of ARP23, which causes local actin remodeling, and then you interfere with E-cadherin stability and cell-cell contact. So we believe that this pathway is operating, and in support of this, we can use drugs that restore RAC1 levels or ARP23, and we suppress the budding. So we believe this is going on. No major shock there. But coming back, we see that both structural and numerical aberrations interfere with this pathway, and yet only the structural ones trigger the budding. And we conclude from this that weakening of E-cadherin contacts between mitotic cells and their neighbors is not sufficient to cause cell budding. There's something else that must be going on. What else is required for budding? When we look at these spheroids or spheres, then they are mosaics generally. Not every cell expresses the centrosome aberration. And we can categorize these spheres roughly by what percentage of cells expresses GFP-NLP. We do this in 4 different bins from 0 to 24, 25 to 29, and so forth. And then we ask, how often do these spheres bud? You might expect to see a linear relationship. The more NLP you have, the more budding you get. But that's not what we see. What we see is a clear threshold. So you have to go above 50% cells expressing, and then you see massive budding, and below you see essentially no budding. And this speaks very much for a cell-cell cooperative phenomenon, not a cell-autonomous phenomenon. Moreover, we see that many budding cells, nearly 50%, do actually not express the NLP. So they don't need to express the NLP. And here are 2 examples. This one gets budding and it does express NLP. You see the green stuff here. And here is another one that buds where you see absolutely the rotation of the spindle like crazy. It goes like this. It's the neighbor that expresses NLP, but not the NLP itself. And we looked more carefully, or Olivier looked more carefully at what these cells, what kind of phenotype they display. And one thing he observed was they're blebbing. You see the membranes here, all these blebs here are stills. they're blebbing like crazy. And this is not a question of NLP overexpression, because in 2D they don't do this, only in 3D they do it. So there's some kind of 3D configuration which causes this blebbing. Moreover, he observes that these cells very often spend a long time in mitosis. Here you see one that does not express NLP, that does not bud, and this mitosis is over very quickly. But this mitosis here, where budding occurs afterwards, this takes 9 hours, so very long time. And that's when it got interesting in the sense that Olivier was scanning the literature and found this study here, which actually was done very close to where we work in Basel and in Zurich. And that is, these people, for whatever reasons, they took dividing cells and put pressure on them. These are biophysicists. That's what kind of biophysicists do. You take a dividing cell and you put pressure on it and you ask what happens. And what they see as they squeeze this poor cell is that a mitotic delay occurs as a function of pressure, and blebbing occurs as a function of pressure. So the exact same thing that we see, blebbing, delayed mitosis. So we teamed up with a neighboring lab in the Biocentrum, headed by Roderick Klim, but the hero is Philipp Oertle. He did all the AFM measurements. So we look now, this is now admittedly in 2D, but we ask, do the mechanical properties of epithelia change when we have structural aberrations, structural centrosome aberrations? And the answer is yes, they do. And you see here, we measure these cells. Here are the blobs. AFM goes over this atomic force microscopy. The cantilever goes over the epithelium. We measure the stiffness of these cells. They're coded here. These guys get stiffer when they express NLP, not when they express numerical aberrations or a control protein. There's a massive increase in stiffness in response to a structural centrosome aberration. So why do mitotic cells bud? Well, because in an epithelium, mitotic cells are actually softer than their interface neighbor. They are the weakest. In isolated, it's the opposite, but this is something I want to go into. So epithelia harboring structural centrosome aberrations show stiffness heterogeneity. So these are the data we have, and here comes now a pinch of wishful thinking and interpretation or speculation added to this. Here is what we think goes on. In a normal epithelium, you have ecadherin, you have cells, this is known, when they go through mitosis, they exert pressure, hydrostatic pressure to their neighbors. And evolution has set up things in such a way that in a normal epithelium, cells are sufficiently deformable to allow dividing cells to settle down within the plane of the epithelium. They just make room for the new neighbors, so to speak. Yeah? Very polite. But in the presence of structural centrosome aberration, first of all, spindle orientation is now randomized because of this ecadherin effect I've told you about. So now you have cells that don't want to go this way, but they actually go this way. And moreover, the cells which express this NLP, they are poorly deformable, and therefore they resist the insertion of the two new cells, and therefore they squeeze out the dividing cell. And now the speculation is, of course, that these cells could have other lesions. They could have, in addition to the centrosome aberrations, they could have oncogenic lesions, any lesion you like, a RAS activation, a MYC amplification, you name it. And I think this mechanism, if it exists, might explain a long-standing conundrum in the field. And the conundrum has always been, why is it that centrosome aberrations are so common in cancer, even though they are expected to be deleterious to cells? They are not healthy. And moreover, you never see that these centrosome aberrations are present in all cells. They always concern only a subpopulation. So now we propose that because we have a non-cell autonomous mechanism, that the dissemination of invasive cells can occur without these disseminating cells necessarily having themselves the centrosome aberrations. It suffices that their neighbor have these aberrations. Okay? So the cells which are kind of sick because they have centrosome aberrations, they may not be the ones that found the next metastasis, if you wish, but they may help squeezing out cells that may carry oncogenic mutations that may not be impaired through centrosome operations. That's what we think. So you may or may not buy this. It's up to you. But we do believe at the moment that structural centrosome aberrations are not just irrelevant byproducts of unbalanced gene expression in human tumors. Instead, they may we know they do cause disruption of epithelial architecture, and they might favor the dissemination of invasive cells through a non-cell autonomous process. And if you want a very simple take-home message, it's right here. This is what we think could be happening if you have these centrosome aberrations, you see? Now, with this, well, I've told you this already. So these are the take-home messages. I've started out by telling you briefly about the kinetochore, in particular focused on this SCA complex, which we believe is both a key substrate and an activator of Aurora B kinase. We found a feedback loop that we think is critical for determining the strength of microtubule kinetochore interaction and hence for error-free chromosome segregation. In the second part of the centrosome, I've introduced a PLK4 still Sus6 module, which we believe stay at the heart of central duplication. I've told you some data that suggests that central amplification, for example, through excess levels of still, may present one prominent root cause of primary microcephaly. And in the end, in admittedly the most speculative part of my presentation, I've proposed to a model whereby structural centrosome aberrations may promote cancer invasiveness based on a tissue culture model for the time being through a non-cell autonomous process. Now, the one thing I regret today is that I could not talk about Lukasz's work, but since he introduced me, you have all seen him. Lukasz was actually a postdoc in my lab. He has done some very nice work. I think Lukasz is happy to be in Brno, and I think Brno should be happy to have Lukasz. And with this, I'm at the end. My lab is now very small. This is Basel. It's a beautiful city, a bit smaller, I understand, than Brno. Here's my funding. This is my lab now. It's very small. This is my wife. This is my dog. And thank you all for your attention.
So thank you very much, Erich, for a really interesting and inspiring talk. So I'm sure there will be some questions. So maybe I start. So what do you think the kind of the NLP aggregate or the blob does to the cell that kind of the amplified centrosomes cannot do? Because you know that they're also able to cluster in mitosis. So in a way, they form some kind of temporal blob. So what's your thoughts on that?
Yes, I think I would be careful at the moment to say that this could never be caused also by amplification. It's just in our model we don't see it. And in our model we believe it has to do with the fact that the microtubule, so you see a microtubule accumulation in both in response to PLK4 excess as in response to NLP excess, but the dynamics are very different. So in the PLK4 case, the microtubules remain very dynamic. In the NLP case, they remain very attached and immobile. And one reflection of this is, for example, when we do a wound healing assay. So we scrape a wound into a confluent monolayer and look how cells migrate to close the wound. The ones that have numerical centrosome aberrations and dynamic microtubule system, they close the wound very rapidly. The NLP ones do not. So for the insiders, and you are, of course, the Pellman work on the invasion through PLK4, and we believe that's a migratory type phenotype. Plus it's collagen 1 dependent phenotype, which I also didn't go into. But there are clearly differences between these two situations. Now, whether they could ever come together, I would certainly be open to that possibility.
Thank you.
Thank you very much for a beautiful talk. Is there any known connection between metabolic stress and centrosome amplification? Because it was shown that under metabolic stress, the sac, for example, is not working properly.
Yeah, I would expect there must be and there will be. You may know about agrsomes. This is when you have misfolded proteins, which is not the same as metabolic stress, I understand, but there are very, very fundamental, if you wish, processes which tend to occur in the surroundings of the centrosome. And then, of course, you have the Golgi apparatus, which always co-localizes with the centrosome. So I believe that these are things which remain to be explored that have been barely touched upon.
Yeah, thank you. I have a question about the centrosome amplification and microcephaly. So you mentioned that the microcephaly actually occurs because the cells cannot divide in the sufficient speed, in a way. But cannot you consider alternative explanation that it's about the asymmetric cell division?
Yes,
because it's quite important.
Absolutely. So what I'm offering here is the most trivial interpretation of much more exciting ones. And once an American told me, when I criticized somebody's work in private conversation with a trivial explanation, he said, "Why do you want to shoot down such an attractive hypothesis with a trivial explanation?" I always think we should first think about trivial explanation, but it's true. So when it first started, when people first recognized that there's a correlation or a very strong connection between centrosome abnormalities and microcephaly, then basically there were three plausible scenario. One was this asymmetric division being impaired. So you change the balance between stem cell pool and progeny, and you deplete the stem cell pool. And that's certainly a very viable hypothesis. The other had to do with migration, because I'm not a neurobiologist, but I understand when you build a brain, you need to have many cells migrate up towards what will be the cortex. Hopefully. And so you could imagine that migration is also impaired if you have a centrosome anomaly. And the third was indeed cell death. And the reason why I now favor cell death is largely because of Renata Basto's work, because in her mouse model where she got this microcephaly induced by PLK4-induced central amplification, she looked very carefully whether she could see evidence for asymmetric division, impairment, and so forth. And to the best of my knowledge, she didn't see very much along those lines. So that brings me back to the most parsimonious interpretation, saying, well, if you kill off cells, then of course you don't build enough cells to build a fully functional brain.
It's maybe a bit naive question, but it's often described that solid tumors are much stiffer than the surrounding tissue. So do you think there could be some connection with the increased cell stiffness you
Yes. Again, this is not my expertise by any stretch of the imagination, but it's my understanding that you have to be very careful about what one talks about between tumor, stroma, all these things. All I can say is this is something, of course, which many people now look at very carefully, because it's become quite fashionable to think about biomechanical properties of not just so we've gone beyond the oncogene/tumor suppressor gene paradigm of tumorigenesis, and now we look at cell-cell interactions and all these things much more carefully. We, the world, not me. But there's one study which I need to mention because it's from our colleagues, Rod Lim's laboratory. They published a paper which apparently had a big impact in the field, where they also using atomic force microscopy, actually looking at biopsies, they report that apparently there's a correlation between softness of certain areas in the tumor and the frequency of metastasis. But there's also a certain interpretation involved, and I'm not sure that this is the final word. But so I think it's hard to say, are they stiff, or it's too simple to say they are stiffer or they are softer. I think it depends on the relative stiffness, softness within a multicellular context that one should ask these questions.
Can I have one more question, maybe a bit speculative? So you showed centrosome as a beautiful structure, and you described the main structural proteins in the centrosome. So do you think we know all the proteins which are built centrosome? And can we one day or soon make centrosome in a tube from the purified proteins?
No. You know, when Lukas knows this, when I was in Martinsried and Matthias Mann, one of the leading proteomics people on this planet, was still in Odense, I thought it would be nice if we knew more about the components. And I had a PhD student at the time, Thibaut Mayor, spent about a year trying to introduce tags into the centrosome so that we could purify them more efficiently. Because as it stands, you start with about something like 10 to the 9 cells, and you end up with 3 microgram of crappy stuff. So this is maybe 70% pure centrosomal protein on a good day, and the rest is junk. And then we always wanted to improve on that. We never managed. But in the meantime, the mass spectrometry instruments got more and more powerful. And so one day, Matthias says, well, let's just try with what you have. Maybe it works. And sure enough, it did. And that led to that paper, which we published in Nature in 2003, I believe, where from a situation where we knew maybe about a handful of, maybe a dozen centrosomal proteins through serendipity, so to speak, all of a sudden we had like, I can't remember, 50 or so novel components. And all these CEP proteins you now read about, most of them have shown up for the first time in that study. And then subsequently, the first author on that study, Jens Andersen, who now has his own laboratory in Odense, he did a second pass with yet better machines, maybe 10 years later, and he had published the paper in EMPO Journal. And there, I would think, if we add it all up, we now come to about 200, 250 proteins in the centrosome. The problem is, how do you define it, and where do you start and where do you end? In other words, there's no membrane around it. So this is, and then you have microtubules that stick out. And so anything with an affinity to microtubules will tend to traffic along the microtubules and may accumulate at the centrosome transiently or with some affinity. So it's very hard where you draw the line, right? And there's a lot of interest now in proteins which transiently associate with centrosomes, maybe for some signaling function. So many people like to think of the centrosome as a solid phase platform to enhance signaling reactions, which would be of interest to you, obviously, yes. And there's a lot of stuff about damage and so forth also relating to centrosomes. I'm quite skeptical, to be honest, on some of these reports. But the idea, of course, is very appealing and very attractive. So if you ask me for a number, I would say probably a couple of hundred, maybe 300, bona fide, genuine. But whether we will reconstitute it, the answer is, the question is what you ask for. So the challenge at the moment is not to make this bowl of spaghetti, which is the PCM, which a lot of coiled proteins, but the challenge is to build the centriole. And there people are really making impressive progress. People like Paul Guichard in Geneva, Paul Gönchi and others, where Pierre Gönchi and others, where they really take purified components from baculovirus or E. coli and then put them together and they start building the cartwheel. The next step probably is going to be to have microtubules to build the centriole. Then the length control is another major issue. We're beginning to learn how to make them extra long or extra short by modulating some proteins, but how it works in vivo, we don't know. It's also a fascinating problem. How do you make sure that every time you build a centriole, you stop at 450 nanometers? Once you have found out how to elongate this thing, why don't you keep going until you hit the wall of the cell? Yeah? But you stop at 450 nanometers. So either you have a ruler, or you have an equilibrium between polymerization and depolymerization that defines the length.
So it seems there are no more questions, so I think we should thank Erich for really, again, for a beautiful talk and also for his visit.
And thanks for the medal, I really appreciate it.